Thanks for inviting me to join this webinar, and today I'm glad to share one of my research. As the title shows, in this work, we proposed a new algorithm named KMAP, Kmer manifold approximation and projection for realizing DNA sequences. So first, let's start with some background. As we all know, the DNA sequence is made up by 4 nucleotides, A, C, G, and T. But this sequence is not random. There are always some patterns rapidly in the genome. A classical example is the transcription factor binding sites. Transcription factor is a protein. It can regulate the gene expression by binding to some special regions of the DNA sequence, like the promoter region and enhancer region. And for one transcription factor, it always recognizes the similar areas of the DNA sequence. So that means the binding patterns can reflect the specificity of different transcription factors. If we can identify their patterns, so it can help us to understand how the transcription factor work together, or they are cooperator or competitors. And it can help us to better understanding the mechanisms of the disease. So in order to pre-process the DNA sequence, the first step is to convert the DNA sequence into small fragments. We will use a sliding window from left to right to cut the DNA sequence, and the sliding window with a fixed length k. So we call these small fragments kmers. And in this work, we only consider the kmer form of DNA sequence. So so under the kmer form, what the pattern of transcription factor binding sites looked like? So we call it a motif. And in a motif, you will see many same kmers, and we call them consensus sequence or consensus kmers. And based on the consensus, you may allow small mutations happen in some seeds. So if you plot the frequency of each nucleotide at each seed, we get a 6 logo like this, and it's a very classical way to represent the binding pattern of a transcription factor. And there are some drawbacks of this realization. Using it ignores the kmer structures in the logo, and you can display multiple motifs in one figure. So that's the back to the definition of a motif. Now we know under the kmer form, within the motif, there are many consensus kmers, and based on the consensus, you may allow small mutation happens, and they are very close to the consensus, but not the same. If we have a bulk of transcription factor binding sites, our task is try to separate them into different clusters, into different motifs. This is the first step. And furthermore, we also hope to have a better visualization of these motifs. And since the motifs in the kmer space, we want to project them into the 2D platform, and we want to project each kmer point we hope to put the motif kmer in the center to form circular pies, and put the random kmer outside to form a ring. So that's our expectation of the visualization part. To achieve this goal, we proposed KMAP, and the whole framework can be divided into three main parts. In part 1, we built a mathematical theory to discuss the Kmer manifold. And this theory is a little bit complex, so due to the time limit, I can't go to more details, but here I just want to introduce the main ideas. So the main idea is given any kmer as an origin kmer, we can put all other kmers on different orbits based on the distance from the origin. Here we use the Hamming distance as a matrix. So that means the Hamming distance will count the mismatch at each seed between two kmer. So that means here we either example we have an origin kmer, and we can put other kmers in different orbits based on the mutation numbers. You have orbit 1, orbit 2, orbit 3 until orbit k. So that means from the origin kmer, you can generate the whole kmer space. So that's the first. So that's the first step. We give a description of the structure of the kmer space. And our task is to try to identify the motif. So if the center kmer considered as the consensus, we need to determine a cutoff of the orbit, so the region can represent the motif, right? So here, what's the cutoff of the motif? And here we explore the distribution of kmer over the orbit, and we found for any key value, we proved this theorem. It's a unimode function, and the number of kmers will increase first and then go down. So the first few seeds, the first few conservative seeds can represent the motif areas because they are very close to the center, and the cancer is not too much. And like if k equal to 8, the radius will be 2. And when k equal to 10 or 11, and the radius will be 3. So that's the definition of the radius. And the kmers within the radius will be called a Hamming ball. So that's the definition of Hamming ball. And now we can go into the part of motif discovery. First, we have many DNA sequence reads, and it contains multiple motif signals labeled by different colors. So first, we will do the kmer count. And the first column is kmer, and the right is their corresponding counts. And for the hair count in the table, it will be considered as a candidate motif kmer. So then we will check their Hamming ball ratio. And if the Hamming ball ratio passed the significant test, it will be labeled by the motif, or it is just labeled by the random kmer. So that's the procedure for motif discovery. And after that, we want to display this motif kmer. And although the kmer space is very large, so we don't want to display the whole space. We just sample representative subsets. And for the sampled kmers, first we will calculate the distance for each pair of them and got a distance matrix. And then we will do a dimensionality reduction method to project the kmer into the 2D platform. So here we, for the dimensional reduction part, we refer to the famous visualization method UMAP. So that's what why we call our method the name KMAP. But we do some changes for UMAP so that it can be suitable for the kmer data. And I will talk about it later, and you will see what kmer data is so special. We conducted 3 kinds of experiments to demonstrate the KMAP's ability in different scenarios. The first is on the high throughput SELEX data. This data comes from in vivo environment, so that means the data is very clean. And in prior, we know there is only one major motif signals in each individual data set. So it's a very nice data set for us to do the benchmark. And here we evaluate the result of motif discovery over 1,200 data sets. And we compare KMAP's result with the SOTA method in this field, MIME. And we define 2 matrix precision and recall. And you will find the precision and recall medians between KMAP and the MIME are 82% and 92%. So then it suggests that KMAP can achieve very similar results with MIME. But I also want to emphasize that KMAP is a lightweight tool, so it's more faster than mine. It's very easy for you to do the motif discovery. And then we go into the motif visualization part. In this part, we compare the visualization result with several classic methods like PCA, MDS, T-SNE, and UMAP. And here the right color represents the motif kmer, and the green color represents the random kmer. As you can see, all these methods have failed. Perhaps MDS and PCA work a bit better because they at least try to group the motif points together. But however, the T-SNE and the UMAP don't even achieve that, even though they are effective in many scenarios. They are powerful tools. So the main reason comes from the peculiar nature of the Kmer space. Kmers using Hamming distance as a matrix, which means the entire space is discrete and finite, right? And you only overturn a limited set of discrete distance ranging from 0 up to k. So that will lead to distance calculation problems and gradient computations. So if you here is a gradient term of the UMAP, you will see there is a term Wi minus Wj square, and since the data contains many same kmers, the constant kmers, so their distance is the distance between them are 0. So that means this term can easily go 0 in the optimization, which will cause gradient exploding. And so here we paid a lot of effort to overcome the problems, and like we add a diffusion term to force the we add a diffusion term to force the 2 points to separate, and also we do a smoothing, and we use a smoothing method to make the distance more varies. So that's here we do some changes of the UMAP framework, and finally KMAP can achieve the goal of our expectation. It puts the motif kmer in the center, and the random kmers put outside. And in the experiment 2, here is about a case study. We want to see how KMAP performs in more real data, and in previous research, researchers have found that ETV6, this transcription factor, plays a key role in the Ewing Sarcoma disease, and the binding pattern of ETV6 is very special. It's a GGA repeat, and a researcher also found that there is another transcription factor called HT-SELEX also bind to GGA repeat. So that means they are competitors. The ETV6 and HT-SELEX co-occupy the GGA repeat binding sets, and when ETV6 occupy this binding sets, and HT-SELEX will be suppressed, which will lead to the Ewing sarcoma. So that's the previous biological research, and here we use KMAP try to confirm this this finding, and we truly found that the FLI1 and the ETV6 binding pattern is GG repeat, and also in some samples the KMAP found other motif signals like AGG repeat. It's also a strong signal, but here we don't know what it means. It's maybe it may represent a meaningful biological signal, but it may also just a false positive signal. So furthermore, we want to explore is there any other transcription factor like FLI1 also be suppressed by ETV6? So we use the ChIP-seq data in the promoter and enhancer region, and use KMAP to do the motif discovery task. KMAP found three new transcription factor, OTX2, BACH1, ERG1, and here is the visualization. And we compare their expression rank, and to see these three transcription factor are in high rank in this area. So that means they might be valuable biological signals. And also the similar situation is in the enhancer is in the enhancer region. We also found OTX2, BACH1, and ERG1, and here we found FLI1. So that means the FLI1 is bound to the enhancer region. And the right figure shows their co-occurrence figure. And you will see these three transcription factor may work together because their co-occurrence frequency over 60%. And last, we use KMAP on the gene editing data. So the gene editing data is generated using the CRISPR-Cas9 technology. In this method, the Cas9 is guarded by RNA that contains the sequence marking the target DNA region. So once the target seed is found, the Cas9 cuts both strands of the DNA. So it will create a double strand break. And the cell's natural repair mechanism will then fix the break. And during this repair process, small insertion or deletion may occur. So that's the base of the gene editing data. So here the reference data, the reference sequence is like the consensus kmers in the KMAP framework, and the gene editing index is just the patterns. We hope to use KMAP to extract the gene editing patterns. So here we have a try, and we use KMAP to detect editing patterns in the AVS1 dataset. And KMAP indeed extracted several clusters. So now we can check these clusters one by one to see what it is. The biggest cluster, the cluster 1, is just most of the reference sequence, and only small errors happened. That means it's maybe the sequence error. And the second largest patterns, there is another patterns and also the reference, but this region is near the 3' end of the reference sequence. So these two clusters is just no special meanings. And then for the class 3, and we found it's just 12 nucleotide deletions based on the consensus. And the next cluster is a 5 nucleotide deletion. Then it's 3 nucleotide insertion, and then the last is 1 nucleotide deletion. So actually, KMAP identified all the top gene editing index in this data sets because this data comes from laboratory, so we know it's ground truth. And so this experiment suggests KMAP might be a powerful tool that can help people to display and extract the gene editing patterns in the data sets. So in summary, KMAP is a computational framework that can for detecting and visualizing DNA sequence motif. And the innovation in this paper is, I believe we are the first paper to discuss, to build the Kmer theory, to discuss the Kmer manifold properties. And by using this theory, we can do the fast motif discovery, and end to end we can achieve a good visualization. And we conducted 3 kinds of experiments to demonstrate that KMAP can be applied into different scenarios. But so far, KMAP still has some drawbacks, like you may see in the more real in the real data, like ChIP-seq data. Since the current model is under the unimode assumption, so it may introduce false positive. And also, we need to extend the Kmer theory from to so that it can deal with the key and the key plus one together. So also we also want to extend KMAP on many other experiments like attack and RNA proteins. So the last is about our team, and this work was completed under the guard of Dr. Lu, and the KMAP paper has been accepted by the Journal of Genome Research. And I'd like to express my gratitude to everyone who contributed to this work. And thanks for your attention. I'm glad to hear your question. Thanks.
Yeah, thank you very much. It's a very interesting talk, to be honest. So I guess there are more questions, but I would like to clarify my understanding. It's a very complex thing for me sometimes. Maybe I don't completely get it.
Okay.
One thing I would like to ask, so when you have this KMAP, you cluster those binding motifs, right? And then those cluster binding motifs, my understanding is like they have the similar sequences, as you showed in your last slides.
Yeah.
But are they the binding motifs from different promote regions of different genes, or they are? So how is that what I understand correct?
You mean the source of the binding seed data?
No, I mean for those clustered binding motifs. For example, in the last slide that you showed about the data set, there you have several clusters, a big one, small one, and the one from the same cluster, they have a very high overlapping of the motifs, right?
Yeah, yeah.
And are these motifs from one gene or for several genes, or how they are identified?
It may comes from several genes. We
So
don't know
from
it
several
from
genes.
the open region of the DNA sequence. So we don't know it contains one gene or multiple genes. We don't know it in prior, yeah. But
Okay.
we only do the clustering task to see the key map can identify one cluster or multiple clusters. Yeah.
Okay. So another thing, you in one of your case study, you mentioned that you identified those two competitive transcription factors.
Yeah, yeah.
Yeah, so they kind of can compete for this one binding site. So biologically, is it possible? Yeah, if you can identify those competitive ones, I mean, how does this work out? I mean, of course, because they have this known transcription binding sites, right? These are already known transcription factors, and their binding sites are already known, right? And
Yeah,
then
yeah.
potentially, they could overlap on this exactly the binding sites that you have identified using your algorithm. So I don't know how is this or how does your algorithm somehow help to identify this. So what's the trick behind this algorithm to identify these overlapping sites? Because from my understanding, if you have a transcription factor, of course you may have several binding sites for one transcription factor, and then you simply can do is like you try to identify whether they have overlapped binding motifs, and then if they have overlapped binding motifs, we can assume they could compete on some binding sites of a particular gene, right? So how does your algorithm do that? That's the thing I don't understand, or I have
Uh,
not got it. Yeah.
Yes, so in the case study, we just want to confirm the previous research. So KMAP can do that. So KMAP just can identify the binding patterns for different transcription factors. So they can tell you the binding pattern of FLI1 is GG repeat. The binding pattern of ETV6 is also GG repeat. But we don't know, is this GG repeat is in the same location? We don't know.
Okay.
Yeah. So you may
So you somehow tell me it's like you identified their similar binding patterns, and then that somehow is in line with the evidence they can overlap or they can compete for binding.
Yeah,
Okay,
correct.
that's the message you want to deliver.
Yeah.
Okay, good. Yeah, fine. That's basically my question. I hope there are more questions from the audience. Do we have other questions? Please feel free to raise your hand or open your mic. And yeah, I think these two talks are really excellent, and they also got very good publications, and that's why we probably could have a discussion with them. That's very nice to have you here. So at least I don't see any questions from the chat, or nobody's turn on the mic or or radio. So I think probably that's all for today, and we would like to thank you again, Mr. Fu. Yeah, and yeah.